My name is Kelsey Martin. My mentor is Dr. Gustafson. My research project is over the location, species, and intensity of intestinal parasites in Missouri wild turkeys. Parasites make up a large percentage of the overall biodiversity of an ecosystem. The wild turkey is a known host to a variety of parasitic helminths species, including cestodes, nematodes, trematodes, and acanthocephalans. These intestinal parasites can affect their health, population dynamics, and evolution. Therefore, understanding the location, prevalence, and intensity of those parasites is essential for wild turkey population conservation and management. Parasites can affect their hosts in a variety of ways, including stealing essential nutrients, causing inflammation, damaging the intestinal mucosa resulting in blood loss, causing reproductive complications, suppressing the immune system, which can predispose the host to secondary diseases, and induce disadvantages during competition for resources such as food and shelter. Currently, there have been no studies on helminths found in wild turkeys conducted in Missouri. My project will provide baseline data on parasite populations in Missouri wild turkeys and assess how the theory of island biogeography relates to host-parasite relationships of wild turkeys and their parasitic helminths. The predictor variables will be defined as host size, host age, and host location. The response variables will be defined as parasite prevalence, infra-population numbers, infra-community structure, and spatial variations of these measurements across the state of Missouri. The theory of island biogeography was first used to analyze and interpret the factors that determine species richness on islands. It has since been applied to symbiotic organisms such as parasite species and hosts. The parasite species richness of a host may be based on the host's size, age, and diet. The geographical location of its environment can also affect the parasite species it comes into contact with. Based on previous studies from the surrounding states, I hypothesize that wild turkeys in Missouri will be infected by 3 cestodes, including Hymenolipus carioca, Metroliaces lucida, and Railiantina williamsi, 3 nematodes, including Ascaridia dismissilis, Capillaria condenflata, and Hetericus gallinarum, and 1 trematode, Echinoperifium recurvatum. To test this hypothesis, I will survey the intestinal tracts of 29 wild turkeys, document which species were present and their abundances within each host. The turkeys were hunter-harvested in the spring of 2019 and submitted to the Missouri Conservation Department staff. Fecal samples were extracted, placed in 70% ethyl alcohol, and transferred to Dr. Gustafson at Missouri Southern State University. Parasites will be removed from samples, stained and mounted on slides, and identified based on morphological characteristics. Cestodes are long tapeworms with segmented bodies. Nematodes display bilateral symmetry with tapered ends. Trematodes are flattened and possess an oral sucker. Acanthocephalans possess a spiked proboscis that they use to attach to the intestinal mucosa of their host. The parasite species will be counted to determine prevalence, intensity, and abundance. Non-parametric statistical tests will be performed to obtain diversity indices and population distributions. This table contains the proposed timeline for my project.